Hi, and welcome to today's Croda Healthcare webinar on HLB, the science behind emulsion stabilization. Thank you for joining us. I'm Liliana Willard, Account Manager for Croda for the Southeast U.S. I encourage you to connect with me and with the rest of our commercial sales and marketing team through the LinkedIn website and live chat links on your screen. Throughout the presentation, we encourage you to interact with us through the survey and Q&A boxes on your screen. We'll be answering questions at the end of the presentation. Samples of the products we discussed today are available free of charge. Please request these from your local sales representative or on our website. Data sheets and additional documentation is available upon request. I'm delighted to be here with Allyson Maron, Application Scientist and today's presenter.
Thanks, Liliana, for the introduction. What do we mean by HLB? HLB stands for the hydrophilic-lipophilic balance. When broken down, hydro means water and philic means loving, so hydrophilic together is water-loving, where lipo means oil, so lipophilic is oil-loving. The balance is the ratio of the oil-loving portion to the water-loving portion. All surfactants must have both hydrophilic and lipophilic portions in order to have surface activity. This will be shown in more detail on the next slide. To explain a surfactant, let's start with a mixture of oil and water, which is immiscible. When the surfactant, seen here as the Croda product, is added to the oil and water, the oil disperses throughout the water and the two become miscible. If we zoom into the oil droplet, the surfactant is seen on the surface of the droplet, showing how we get the name surfactant. Surface, active, agent. Looking at the surfactant, the head, as shown by the blue circle, is in the water, so that is the water-loving or hydrophilic portion, where the tail is in the oil droplet, so that is the oil-loving or lipophilic portion. The Hlb system was created as a tool for formulators to make emulsions. The basics of the system are that non-ionic surfactants each have an Hlb value between 0 and 20. This does not apply to anionic, cationic, or amphoteric surfactants. This Hlb value correlates to the surfactant's expected solubility in oil or water. If the Hlb value of your surfactant matches the application or formulation requirement, which I will explain more about later, then you will get good surfactant performance. The actual Hlb values, what do they mean? Scales are used in the figures to help demonstrate the differences in Hlb values. As seen in the figure on the left, if the Hlb value for the surfactant is below 9, then there are more oil-loving groups than water-loving groups, and the surfactant is lipophilic. If the Hlb value for the surfactant is above 11, as seen in the figure on the right, then there are more water-loving groups than oil-loving groups, and the surfactant is hydrophilic. If the Hlb value is between 9 and 11, then the water-loving and oil-loving groups are balanced, as shown in the figure in the middle. Lipophilic surfactants with Hlbs below 9 are used for water and oil emulsions, while hydrophilic surfactants with Hlbs above 11 are used for oil and water emulsions. So why should you use the Hlb system? It uses a systematic approach when developing emulsions. Not only non-ionic surfactants have Hlb values, many oils have required Hlb values. By knowing the required Hlb of your oils, the appropriate emulsifier, another name for a surfactant, can be chosen for the emulsion. By knowing the required Hlb, a combination of emulsifiers can be used to closer match the Hlb and to possibly give a more stable emulsion, as the surfactants are more tightly packed around the oil droplet. This systematic approach will ultimately save ingredients and time. There are some limitations to the Hlb system, though. The ratio of oil phase to water phase is not considered, nor is the amount of emulsifier required in the emulsion. The Hlb theory does not account for liquid crystal formation, water phase thickeners, synergy between ingredients, the structure of the emulsifier, or the use of fatty alcohols. As stated before, Hlb does not work with ionic materials or emulsifying waxes. temperature of the formulation and salts are not considered for ethoxylated materials. Lastly, the Hlb theory does not indicate the best emulsifiers for water and oil emulsions. In previous slides, I have explained Hlb values and mentioned required Hlb. I will now explain the difference between the two. To recap, the Hlb value is given to non-ionic surfactants and is between 0 and 20. Every lipophilic ingredient or every ingredient in the oil phase has a required Hlb. These can be found in our Hlb booklet. Please contact your Croda sales representative for a copy. By knowing the required Hlb of a single oil or your oil phase, emulsification can be achieved with the least amount of the right surfactants. The required Hlb is determined by a simple experiment, while the Hlb value for the surfactant can be calculated. The Hlb value can be calculated theoretically or practically. Theoretically, the Hlb of non-ionic ethoxylates is determined by the weight percent of ethylene oxide divided by 5. Ethoxylates are ingredients made from reacting a known amount of ethylene oxide with another chemical. The weight percent of ethylene oxide is chosen as this makes up the hydrophilic portion of the surfactant. This is divided by 5 to keep the Hlb scale smaller and more manageable. The next slide will show an example of how to calculate this for a known non-ionic ethoxylate. Practically, the Hlb can be determined experimentally. For our example Hlb value calculation, we will be using Bridge O20, which correlates to the inky name oleith 20. This product is a 20 mole ethoxylate of oleal alcohol. First, the molecular weight of the ethylene oxide is calculated. Having 20 moles of ethylene oxide, 20 is multiplied by 44, which is the molecular weight of 1 mole of ethylene oxide, to give 880. This number is added to the molecular weight of oleal alcohol, 270, giving 1150 as the molecular weight of Bridge O20. The weight percent of the EO is found by dividing 880 from the total molecular weight of Bridge O20, 1150. Lastly, the weight percent is divided by 5, as per the equation, to give us 15.3, the Hlb value of Bridge O20. A video will now begin showing experimentally how to find the required Hlb for an oil. hope the video helped pull together some of the concepts we have already covered. Now we will look at how the Hlb value can correlate to the application requirement. For an application of compatibilizing dissimilar oils, very lipophilic surfactants with an Hlb from 1 to 3 are required. Water and oil emulsions require surfactants with an Hlb of 4 to 6, which are lipophilic. For wetting powders into oils, a surfactant with an Hlb of 7 to 9 is required. Surfactants with an Hlb from 8 to 12, closer to an even balance of lipophilicity and hydrophilicity, are required for oil and water emulsions. Very hydrophilic surfactants, Hlb 13 to 15, are required for detergent solutions. These correlations are based on long time experience of using Croda emulsifiers, but exceptions do exist. For example, a few excellent detergents have been found in the Hlb range of 11 to 13. From the previous slide, Croda has a range of surfactants that will create whichever application you are looking for. SPAN 85 and Crodesta F10 can be used for compatibilizing dissimilar oils. For water and oil emulsions, we offer SPAN 80, Cithrol GMS, Bridge S2, and Bridge O2. SPAN 20 can be used for wetting powders into oils. For oil and water emulsions, our products in the Hlb range of 9 to 12 include Merge S8, Crodesta F110, and Bridge O10. Our Glycerox 767, Crodesta F160, and Tween 80 are in the Hlb range for detergent solutions. These are some examples if you are planning on only using one surfactant. If you were to combine surfactants, 50% SPAN 80 and 50% Tween 80 give an Hlb of 9.6 and could be used for an oil and water emulsion. Now I will go over how to calculate the required Hlb for a formulation. Let's look at the formulation and determine the oil-soluble ingredients, not including the emulsifiers. Next, you find the weight percent of each of the oil-soluble ingredients by adding them together and dividing each by the total amount. Multiply the weight percent of each ingredient by its required Hlb. Add these values to get the required Hlb for your formulation. Remember that all oil-soluble ingredients need to be considered when calculating Hlb. By adding one ingredient to a stable emulsion, this can change the required Hlb of the entire system. On the next few slides, we will go over a sample formulation and how to find the required Hlb. On this slide, we have our sample formulation. In the yellow box, we have the oil phase, comprising of 20% of the formulation. In the blue box is the water phase, comprising of 80% of the formulation. For calculating Hlb, we will take the oil-soluble ingredients into account, which for this formulation include mineral oil, cetyl alcohol, also known as Croda's Crodacol C95, caprylic capric triglycerides, known as Croda's Crodamol GTCC, and isopropyl isostearate, Croda's Crodamol IPIS. From the previous slide, the percentage of mineral oil was 10, cetyl alcohol was 4, and caprylic capric triglycerides and isopropyl isostearate were 3. When added together, the total oil phase equals 20%. To find the weight percent of each ingredient, the percentage of each ingredient in the formulation is divided by the total of all oil-soluble ingredients, 20. The weight percent of each ingredient comes out to 50% for mineral oil, 20% for cetyl alcohol, and 15% for both caprylic capric triglycerides and isopropyl isostearate. The contributions of each ingredient are then multiplied by the ingredient's known required Hlb. These values can be found in Croda's Hlb booklet. Please contact your local sales rep if you would like a copy. The column Hlb in formulation shows the Hlb of each ingredient in the formulation while taking into account the percentage of ingredient. From there, the required Hlbs of each ingredient are summed to give the required Hlb for the formulation, which for this formulation is calculated as 10.8. Now that we know the required Hlb of the formulation is 10.8, a surfactant or surfactant combination with the same Hlb value should be used in the formulation to get the desired emulsification. Some possible surfactants and combinations to achieve an Hlb of 10.8 include 100% Merge S8, PEG 8 stearate, a combination of 40% span 80 sorbitan oleate and 60% tween 80 polysorbate 80, or a combination of 55% bridge S20 stearth 20 and 45% bridge S2 stearth 2. To get the best possible emulsification, here are some highly suggested combinations of emulsifiers which are known to work well together. Span 60 and tween 60. Both are on the European Pharmacopoeia and are used hot process. Span 80 and tween 80. Both are on the European and U.S. Pharmacopoeias and are used cold process. And lastly, bridge S2 and cetamacrolol 1000 BP. Both are on the European and U.S. Pharmacopoeias and are to be used hot process. In summary, Hlb is a number system used to show how oils and surfactants will likely interact. Non-ionic surfactants have Hlb values. The higher the value, the more hydrophilic or water-loving the surfactant. The lower the value, the more lipophilic or oil-loving. Oils and formulations have Hlb requirements. By matching the Hlb value with the Hlb requirement, the application will perform well. And finally, Hlb is not rocket science, but it is a very useful and time-saving guide.
Thanks, Allyson. This is a very high-level presentation. If you are new to Hlb or need more details, please contact your sales rep to set up a meeting. Croda is always open to new opportunities to develop innovative excipients. If you are facing drug development challenges with your current excipients or are interested in collaborating with our new product development or applications research and technology teams, please don't hesitate to let us know by submitting a request in the survey box to your left or through our emails listed below. We'd love to help you find a solution. If you have specific questions or product inquiries for Allyson or our sales team, we encourage you to follow and connect with us on LinkedIn. Be sure to take advantage of our new live chat functionality and register on our website to opt in to receive future notices about upcoming webinars, product launches, and market insights. We'll now open up for questions and we'll continue to answer questions from the Q&A box. Okay, we have our first question. Is Hlb only applicable to healthcare applications?
Thanks, Lily. No, Hlb is not only applicable to healthcare, it is applicable to anything or any application that uses an emulsion.
Thank you. Do you have the Hlb value of sunscreens?
So for this question, I'm guessing it's the pigments. I'm not exactly sure what they mean by sunscreens, but if it's the pigments, then there is no Hlb for that because those should not be oils.
Thank you. Can you please explain why it is better to use a combination of high Hlb and low Hlb?
Sure. So with high Hlb, the product is more hydrophilic, so the water head of the surfactant will be bigger. And with low Hlb, it's more lipophilic, so the head will be smaller. So when you have these surfactants surrounding your oil droplet, all the heads are going to be in the water and the tails will be in the oil. Now, if you have all hydrophilic heads, so all high Hlb, then you'll have spaces in between the surfactants. So to fill in those little spaces and for the interfacial tension on the oil droplet, you want to fill it in with the smaller heads. So that's why you would use a high Hlb and a low Hlb.
Thank you. That was a great explanation. Why is the Hlb scale applicable only for non-ionic surfactants?
So it's only applicable for non-ionic surfactants because we're not taking into account a charge.
Okay, great. Thank you. Can the experiment be performed for a combination of multiple lipophilic ingredients?
Yes, it can. So if you would just put your ingredients in at the same percentage as what your formulation would have, and you just test your emulsifier, you will get the same separation as if you only had one lipophilic ingredient.
Thank you. Could you use the Hlb kit to test the required Hlb for the whole oil phase instead of testing individual oils?
Yes, you can. You just have to make sure that your percentages are the same as what's in your formulation.
Are there any physical tests to determine Hlb besides the Hlb kit from Croda? Is there a general rule of thumb for Hlb values for fragrance oils?
So physical tests, I don't know of any besides our test kit. And for the general rule of thumb for oils, I am not aware of that. We don't use fragrance in healthcare, but I can, if you have a question, I would contact your sales rep, your local Croda sales rep, and they should be able to connect you with a technical person on the personal care side.
Thank you. What should we do for anionic surfactants? How should we proceed?
You would not be using Hlb. I think it's with that, it's more, you would unfortunately just have to test in the lab.
Would you happen to know which surfactants are approved for parenteral use and what their Hlb values are?
Yes. So if you contact your local Croda sales rep, we have our Croda Healthcare Guide, which will give you all the approved uses for our products and their Hlb values if they have one.
Thank you. Can you please explain why some newer non-ethoxylated emulsifiers, such as polyglycerols, do not have Hlb values?
At the moment, I don't have an answer to that, but I can get back to you on that.
Okay, thank you. Could inclusion of silicone have any effect on required Hlb?
Yes. So anything in your formulation besides for the oils will have an effect on the Hlb, and well, not in the Hlb exclusively. It'll have an effect on the stability. So silicone shouldn't have an effect on the Hlb, but it should have an effect on the stability, and that's where you have to be careful and do some trials in the lab.
Okay, thank you. Do you know the molecular interaction between SPAN 80 and polysorbate 80?
Yes, but this is something that we would have to go into later. So if you contact your sales rep, you can be in touch with a technical person who can talk to you about this.
Is Hlb only applicable to non-ionic emulsifiers?
Yes, it is.
Is there a good way to calculate theoretical Hlb for non-ionic surfactants from only its chemical structure?
Yes, so depending on the structure, so in the presentation for ethoxylates, it would be the percent of ethoxylate in the chemical divided by 5. So depending on what your structure is, there are different equations. So if you contact us, we would be able to help you with that.
Okay, thank you. How does the formula apply to non-ionic surfactants that are based on polymers? As they have different, oh, sorry.
No, go on. I'll finish, please.
Sorry, I'll repeat that. How does the formula apply to non-ionic surfactants that are based on polymers?
So as I stated in the limitations, there is limitations for polymers and for certain waxes of the Hlb.
Okay, thank you. How do anionic surfactants have Hlb values of 40?
So I feel like this is a long answer, but there are There are different HLBs that go higher, but for Crota and for our HLB, we only do 0 to 20, and there are some people that try to get HLB values of other surfactants, but we don't use those HLB values.
Okay, thank you. Drugs have their own log P values. How can we use the log P value of a drug with Hlb of carriers?
So for this, Hlb doesn't really take into account your active. So it more takes into account just the emulsification of your application. So once your application, you know it will become an emulsion, that's where you can then add in your active. And especially with, just because your Hlb is 12 doesn't mean that every product that has an Hlb of 12 will give you a stable emulsion. So that's where you have to go into the lab and try different combinations. And if the, tween and span, that family doesn't work, maybe you need to look at bridges. So it's, unfortunately, once you have your Hlb value, you still need to go into the lab and try out different combinations to see what is going to make the most stable emulsion.
Thank you. Would you happen to know the required Hlb for silicones or silicone alternatives?
I do not off the top of my head, but if you contact your sales rep, we can try to find that for you.
Thank you. How do you determine the right Hlb of a single oil?
So for that, you would use our test kit, and you would show, do the experiment that was shown in the video, and you would just put the single oil in with the different Hlb oils, emulsifiers, and see which one emulsifies the best.
Thank you. How would you improve wettability of product that already contains a high and a low Hlb surfactant?
So for there, it would then depend on what else is in your equation, is in your formulation, excuse me. Or you could also try, if you want to improve your wettability, maybe you need to try different surfactants.
Can we calculate the Hlb based on chemical structure?
Yes, you can. Depending what the chemical structure is, it'll give you, it's a different equation, but if you contact us, we should be able to help you with that.
Can you use only non-ionic surfactants as the emulsifier in lotion or a cream?
No, you can use any surfactants in lotions or creams. It's just if you do that, then you wouldn't use the Hlb system to get your emulsification properties.
Thank you. Is there an Hlb value for emulsifying CBD?
That's something that we would have to take offline. If you could contact your sales rep and you could get in contact with the technical person, we could answer that question.
Thank you. How do you determine the Hlb of a combined surfactant?
So combined surfactants is similar to finding the required Hlb of your formulation. So you would take the percentage of your emulsifier and then multiply it by its known Hlb value and do the same for however many emulsifiers you want to use. And then from there, you would divide it by the total amount and you would get how much of each emulsifier you need to add to get your Hlb.
Okay, thank you. If there are no ethoxy groups, how can we calculate the Hlb value?
So there are other equations if there are not ethoxylated groups, but those, again, it is in the booklet, excuse me. So if you get our healthcare booklet, there should be other equations in there.
Okay, thank you. Why is low Hlb needed for water in oil emulsions?
So low Hlb is for lipophilic emulsifiers. So lipophilic emulsifiers are more likely to be used in water and oil emulsions. But it's not a fast and hard rule. So if you do find that you have a high HLB and it still creates a water and oil emulsion, then that's just an exception to the rule.
Okay, thank you. If you run through the Hlb test and nothing makes a good emulsion, should you try a different base chemistry, or is there a better route?
So if nothing makes a good emulsion, then what you want to do is you want to find the Hlb that basically separates or is unstable last. And then from there, I would try that Hlb, because there are times where none of them actually create a stable emulsion. But if there's one like Hlb 12 takes longer than the others to separate, then that will be your Hlb value.
Okay, thank you. Is there any relationship between toxicity and Hlb value?
No, not that I know of.
Which are more suitable emulsifiers, non-ionic, for oil and water emulsions for injectable applications?
I don't have specifics off the top of my head, but again, if you contact us, we should be able to get you a list of suitable emulsifiers.
Okay, it looks like we've answered pretty much all of the questions. Some of the questions that I did not read out loud have to do with copies of the presentation, and you will receive a link within a couple of days that will take you to the on-demand version of this presentation. There also is a video clip available on YouTube that is for the video clip we watched here today. If you'd like to get a copy of that link, please contact your local Croda sales representative and they can get that out to you. Thanks again to everyone for joining us. If you have any further questions, please reach out to us personally or visit the live chat feature at crodahealthcare.com. Thank you so very much and have a wonderful day.